Welcome back, everyone. Today, we're diving deep into canine cancer immunotherapy.
Oh, this is going to be fascinating.
It is. It's amazing how much companion dogs are contributing to the advancement of this field.
Not just for themselves either.
Right, for humans, too.
Absolutely.
So we're going to be looking at this review article from Clinical Cancer Research.
Yeah, by Brian and Mates.
Yes, Brian and Mates.
It is a dense one, lots to unpack.
It is. But that's what we're here for, to break it all down. So first things first, why dogs? What makes them so important for cancer immunotherapy research?
Well, think about it. Dogs share so much with us.
Our environments.
Our environments, our lifestyles.
Sometimes even our food.
Exactly. So they're exposed to a lot of the same carcinogens that we are.
Which means?
They develop many of the same cancers.
Oh, wow.
And often at higher frequencies than humans.
Really? Like what kinds of cancers?
Osteosarcoma, you see that a lot. Hemangiosarcoma, lymphoma, the list goes on.
So that makes sense, right? If we want to study these cancers and how to treat them, dogs are a really valuable model.
They really are. Plus, there's another interesting factor, the lack of rigid standards of care in veterinary medicine.
Okay, now that's a bit counterintuitive. How does that help research?
Well, think about it. This flexibility actually allows for faster accrual in clinical trials.
I see.
There's more room to try different combinations of treatments, explore earlier line therapies.
Because in human medicine,
You have those strict protocols.
which are important, obviously.
Crucial for safety, but they can also slow down the research process.
So it sounds like this flexibility in veterinary medicine really speeds things up.
It does. And when you combine that with dogs' complex immune systems,
And
the diverse genetic makeup of different breeds,
easy you come.
the most realistic models for testing these novel immunotherapy approaches.
Okay, so we've got this perfect storm of factors that make dogs incredibly valuable for this research. They get the same cancers, flexible treatment protocols, complex immune systems, genetic diversity. It's like they're the ideal test subjects.
In many ways, they are.
So let's talk about some specific types of immunotherapy. The article starts with nonspecific immunostimulants. Can you break that down for us?
Sure. Basically, these are ways to just kickstart the immune system,
Kickstarted into doing what?
attacking tumor cells. But without targeting specific antigens, it's like giving the immune system a general boost, making it more vigilant and aggressive.
Okay, I'm following. And the article highlights a really cool success story with a molecule called LMTPPE.
Oh, yeah, that's a fascinating one.
Tell me about it.
So LMTPPE, it's a derivative of MDP, and it activates these immune cells called monocytes and macrophages, makes them much more efficient at attacking tumor cells.
That's awesome. But how does it actually get to the tumor cells?
Well, that's the clever part. It's encapsulated in liposomes.
Liposome.
Tiny little fat-like bubble.
Okay.
And those help it reach the tumor more effectively.
So it's like a targeted delivery system.
Precisely.
That's pretty ingenious. And the article says that this therapy was really successful in dogs with osteosarcoma.
It was a game changer, really. A pivotal moment in canine human translational research.
Tell me more.
Well, back in 1989, a study by McEwen and his colleagues showed that dogs with osteosarcoma who received LMTPPE after surgery.
After surgery?
Yep, after surgery. They had a significantly longer time before the cancer spread.
Oh, wow.
And they lived longer overall compared to dogs who just got a placebo.
So right from the start, this treatment was showing some serious promise. What happened next?
Well, that initial success, it led to another study in 1995.
Right.
Kurzmatellen and his colleagues, and in this study, they tested LMTPPE alongside cisplatin,
Cisplatin.
a standard chemotherapy drug.
Got it. So did that combination therapy work?
That's where things get really interesting. They found that giving the LMTPP after the cisplatin worked beautifully. Giving them together, not so much. Didn't seem to have much benefit at all.
That is strange. What could explain that?
Now, that is the million-dollar question. One theory is that giving the two treatments at the same time might somehow interfere with the immune-boosting effects of LMTPPE.
That's a real head-scratcher. It shows how much we still have to learn about the intricacies of the immune system.
It really does. But here's the amazing part. Those dog studies, they directly led to human trials.
No way.
Yes. And the results were so good that LMTPPE is now approved for chemoimmunotherapy in both Europe and South America.
Wow, that is a fantastic example of how research in dogs can directly translate into life-saving treatments for humans. Talk about One Health in action.
It's a beautiful thing.
Okay, so that's LMTPPE, a real success story. Now let's move on to another type of immunotherapy. vaccines.
All right. And the review article, it differentiates between autologous and allogeneic vaccines.
Can you remind us about the difference?
Of course. So, autologous vaccines, they're made using tumor cells taken from the individual dog being treated.
Okay, personalized.
Very personalized. While allogeneic vaccines, they use tumor cells from a different dog, kind of like a one-size-fits-all approach.
Got it. So have these vaccine approaches been as successful as LMTPPE?
Sadly, not quite. The results with both autologous and allogeneic vaccines have been a bit mixed.
Oh, that's too bad. Similar to what they've seen in human trials, I guess.
Yeah, unfortunately, very similar.
Well, are there any specific examples of autologous vaccines that have shown any promise?
Yeah, there have been a few glimmers of hope, like, for instance, some studies on canine meningioma,
Meningioma.
a type of brain tumor.
Okay.
And these studies have shown that vaccines can actually induce an immune response against the tumor.
So the immune system is recognizing and attacking the tumor cells.
Yes. There's also been some interesting research on lymphoma vaccines,
Lymphoma vaccines.
potentially increasing survival times in dogs with lymphoma.
That sounds encouraging, but it sounds like the success has been limited so far.
It has, yeah. And while there are some commercially available autologous vaccines popping up,
But really?
yeah, more research is definitely needed to figure out their long-term efficacy, how to make them work consistently.
Okay, so the jury's still out on autologous vaccines. What about allogeneic vaccines? Anything promising there?
Again, the results are mixed. There was some initial success with melanoma and hemangiosarcoma cell line vaccines,
Okay.
but no commercial allogeneic vaccines available yet.
So it sounds like both types of vaccines are still in those early stages of development.
They are, but, and this is exciting, there are some really cool new avenues being explored right now.
Okay, tell me more.
So, for example, researchers are using Salmonella bacteria.
Salmonella.
Yeah.
Like the food poisoning bacteria.
Yep, that one. They're using it to stress out tumor cells.
Stress them out. How?
It makes them more recognizable to the immune system. Essentially, they're using bacteria as a tool to boost the immune response.
That is fascinating. So they're turning bacteria into an anti-cancer weapon.
That's a great way to put it. And there's also some interesting work being done with cryopreserved listeria.
Listeria.
It carries the HER2 gene.
And that could be a game changer.
If it proves to be safe and effective, absolutely.
Okay, so bacteria-based vaccines are definitely something to watch. What else is on the horizon?
Well, there's also been progress with DNA and peptide vaccines, and those are promising because they're relatively cheap to produce,
Oh, that's good.
and they could be really beneficial additions to standard care.
Any specific examples?
Yeah. There's a USDA-approved DNA vaccine that targets human tyrosinase.
Human tyrosinase.
It's a protein, and it's found in canine melanoma.
Got it.
And this vaccine, it's been shown to extend survival times significantly. A big win.
That's fantastic.
And then there's also an EGFR peptide vaccine.
EGFR.
It's another protein.
Okay.
And this one's showing remissions in dogs with metastatic osteosarcoma.
Wow. So it sounds like these newer vaccine approaches offer a lot of hope.
They do. It's an area of very active research. And canine models, they're playing a key role in pushing the boundaries of what's possible.
This is all so incredible. I'm learning so much. Are there any other types of immunotherapy being researched in dogs?
Oh, tons. But one of the most exciting and rapidly evolving areas involves immune checkpoint inhibitors.
Okay, refresh my memory on how those work.
Immune checkpoints. Think of them as brakes on the immune system.
Brakes.
Yeah, they prevent it from overreacting.
Attacking healthy cells.
Exactly. But cancer cells, they're sneaky.
Nikki, how?
They can exploit these brakes to hide from the immune system, evade detection.
Oh, wow, I see.
So immune checkpoint inhibitors, they release those brakes.
And allow the immune system to recognize and attack the cancer cells more effectively.
Exactly. And they're already making waves in human oncology, and now they're showing huge potential in dogs, too.
That is so cool. Are they working in dogs?
Well, the good news is that they've identified those key checkpoint molecules in canine tumors and antibodies against canine PD-1 and PD-L1.
Those are two of the most important checkpoint proteins, right?
You got it. Those antibodies, they've already been developed.
That's amazing. And what have the early trials shown?
Encouraging results. There's evidence that these checkpoint inhibitors are effective against melanoma,
Okay.
sarcoma, and lymphoma in dogs. It's still early days, but the potential benefits are enormous.
So it sounds like these checkpoint inhibitors could really revolutionize canine cancer treatment. Are there any downsides, concerns?
Well, just like in humans, there have been some immune-related side effects observed in the trials.
Yeah, that makes sense. You're essentially unleashing the immune system, so there's always a risk of it going a little overboard.
Exactly. But the key is figuring out how to strike that balance.
Between effectiveness and safety.
You got it. And canine models are helping us do just that. They're providing valuable insights into how to use these powerful therapies safely and effectively.
Okay, so we've covered non-specific immunostimulants, vaccines, and checkpoint inhibitors. What other strategies are being explored to break immune tolerance?
Oh, there's a whole bunch. Researchers are looking at all sorts of approaches, like using nebulized IL-15.
Nebulized, so like inhaled.
Exactly. It's shown some promise against lung metastasis in osteosarcoma. Really targeted approach, delivering the treatment directly to where it's needed most.
That's fascinating. What else is in the pipeline?
Well, there's also the use of two drugs, losartan and toshrenib.
Okay, and what do those do?
They block a specific molecular pathway.
Involved in.
Cancer spread, it's like cutting off the cancer's escape route.
I like that. Any other promising approaches?
Oh, yeah, there's the use of tumor-tropic viruses, like VSV, IFMA-NIS, and those have been shown to increase survival times in dogs with osteosarcoma.
So many different approaches. It's almost overwhelming.
It is a lot to keep track of. But the main takeaway is that this research in canine models, it's paving the way for some incredibly exciting new treatments.
And what we learn in dogs can directly benefit human cancer patients.
Absolutely. That's the whole point.
That's really the heart of this whole deep dive, isn't it? The incredible translational potential of canine cancer research.
It really is.
And the next frontier in this field, cellular therapies.
Oh, yeah. That's where things get really wild.
Now that sounds intense. Let's dive into that next.
So we're talking about using living cells as weapons against cancer.
That does sound intense. What kinds of cells are we talking about?
We're talking about harnessing the power of the immune system. T cells, natural killer cells, those kinds of cells that directly target and destroy cancer cells.
Wow, that's amazing. It's like you're taking the immune system's best soldiers and just sending them into battle against the tumor.
That's a great way to think about it. And the article dives into quite a few different types of cellular therapies that are showing promise.
Okay, like what?
Well, one early example is using this human cytotoxic T cell line called TAL-104.
A human cell line. So they used human immune cells in dogs.
They did. And you might be wondering how that even works.
I am, actually.
It's because these TAL-104 cells are what we call MHC non-restricted.
MHC non-restricted. Okay, break that down for me.
Sure. So MHC stands for Major Histocompatibility Complex. It's basically a set of proteins on the surface of cells that helps the immune system tell the difference between self and non-self.
Okay, that makes sense.
And usually, immune cells need to match the MHC of their target cells to recognize and attack them.
So it's like a lock and key system.
Exactly. But TAL-104 cells, they don't have this restriction. They can kill target cells regardless of their MHC type.
So they have a universal key that can unlock any door.
That's a good analogy. And while the early studies with TAL-104, they didn't actually lead to a widely used therapy.
They didn't.
No. But they provided valuable insights that paved the way for other cellular therapies, especially the ones using a dog's own immune cells.
So it was a proof of concept.
Exactly. Kind of laid the groundwork for what came next, activated autologous T cells.
Autologous meaning?
Meaning they come from the dog itself. So you take the dog's own T cells, extract them, activate them, and then reintroduce them to target their own tumor.
Oh, wow, that is amazing. And has that been successful?
It's been remarkably successful in dogs with osteosarcoma. There was one study where dogs received these activated T cells after surgery to remove their tumors, and the results were astounding.
Like what kind of results?
Long survival times, even spontaneous remission of metastases in some cases.
Spontaneous remission? Wait, so your cancer just disappeared on its own?
In some dogs, yes. It was truly remarkable.
That's incredible. What happened after that study? Did that treatment become available?
It did. That study led to the commercialization of this therapy by Elias Animal Health. It's actually currently offered as an experimental treatment, and the results have been so promising.
So promising that.
It received a fast-track designation for a human glioblastoma trial by the parent company, TVAX Biomedical.
Wow. So from dogs to humans. And expedited approval for a human trial. That is translational research in action.
It really is.
Okay, so TAL-104, activated autologous T cells. What other kinds of cellular therapies are being explored?
Well, another cell type showing promise are natural killer cells, or NK cells.
NK cells, what are those?
They're part of the innate immune system, kind of like the body's first line of defense. And unlike T cells, which need to be trained to recognize specific antigens,
NK cells are just ready to go.
always on patrol,
Oh.
ready to attack anything that looks suspicious.
Oh, wow. So they're like the immune system's rapid response team.
Love that analogy. And one of their specialties is recognizing and eliminating cells with reduced MHC-I expression.
MHC-I, that's that major histocompatibility complex, right? The self versus non-self thing.
You got it. And some cancer cells, they try to evade the immune system by basically downregulating their MHC-I expression, trying to make themselves invisible.
But NK cells can see through that.
They can. And the article mentions that combining radiation with NK cell injections has shown some promise in dogs with osteosarcoma.
Interesting. So the radiation helps boost the NK cells' activity.
It seems to. Makes it a pretty potent 1-2 punch.
Okay, so we've got TL104, activated T cells, NK cells. Anything else?
There's also been exciting progress in developing allogeneic INK cells.
Allogeneic INK cells.
Invariant natural killer cells. They are specifically reactive to certain tumors.
Invariant. What does that mean?
It means they have this specific type of T cell receptor that recognizes a particular type of antigen, one that's often found on tumor cells.
Oh, okay.
And because they're allogeneic, meaning they can come from a donor, it opens up the possibility of creating off-the-shelf INK cell therapies.
Oh, wow. So these INK cells are like highly trained assassins programmed to take out specific targets, and having them readily available would be a game changer for treatment.
Exactly.
Yeah.
And that brings us to one of the most cutting-edge areas in cancer immunotherapy. Not just for dogs, but for humans, too. CAR T cells.
CAR T cells. I've heard that term thrown around, but I'm not really sure what it means.
CAR T stands for chimeric antigen receptor T cells. They're essentially T cells that have been genetically engineered.
Genetically engineered to do what?
To express this special receptor, the chimeric antigen receptor, or CRR, and that allows them to target specific tumor antigens.
So they're like superpowered immune cells designed to seek and destroy specific cancer cells.
Exactly, and it's one of the hottest areas of immunotherapy research today. There's already been some promising early work in dogs with B-cell lymphoma.
Oh, really? What have they found?
Well, they've been able to successfully engineer CAR-RT cells to target CD20. It's a protein found on B-cell lymphoma cells.
And is it working?
In one dog, they even observed cytokine release syndrome.
That sounds serious.
It can be, but it's manageable. It's actually a side effect that can occur with CAR-T therapy.
Okay, so it sounds like they're making progress.
They are, but there are definitely still challenges. But the potential of CAR therapy, it's enormous. And canine models, they're helping us refine this technology, make it safer, more effective.
What other types of CAR T cells are they developing for dogs?
Well, researchers are working on CAR T cells that target B7H3.
B7H3.
It's a protein found on osteosarcoma cells. And there's actually a company called Life Engine Laboratories.
What about them?
They're developing a really innovative approach using gene-edited xenogeneic CAR T cells.
Xenogeneic, does that mean they're using CAR T cells from a different species?
You got it. They're taking CAR T cells from another animal, like a pig, and genetically modifying them so they can be used in dogs. You can actually see a diagram of this process in figure 3 of the article.
Wow, that is some next-level genetic engineering. So what's the benefit of using these xenogeneic CAR T cells?
Well, it could be a real breakthrough in CAR T manufacturing, making it easier and cheaper to produce large quantities of these cells.
That would be amazing, making this therapy more accessible.
Exactly. And research in dogs is helping to make that happen.
Okay, we've talked a lot about immunotherapy, but cancer treatment isn't just about immunotherapy, right? What about traditional therapies like radiation?
Great point. And actually, one of the fascinating things about this research is the discovery that radiation therapy, it's not just about local control anymore.
What do you mean by that?
Well, traditionally, radiation therapy has been viewed as a local treatment. You zap a tumor with radiation to shrink it or destroy it at that site. But there's more and more evidence that radiation can also have systemic effects.
Systemic, so affecting the whole body.
Yeah, it can actually influence the immune response to tumors throughout the body.
Wait, really? So you're saying zapping a tumor with radiation can somehow boost the immune system's ability to fight cancer cells that have spread to other parts of the body.
That's exactly what the research is suggesting. It's kind of a paradigm shift in how we think about radiation therapy.
That's incredible. But how does that even work? I thought radiation killed cells, not boosted the immune system.
It's a really fascinating question, and researchers are still trying to figure out exactly how this all plays out. But one theory is that when delivered in a specific way, radiation can trigger what's called immunogenic cell death.
Immunogenic cell death. That sounds pretty intense.
It is. It basically means that the cancer cells, when killed by radiation, release certain molecules. And these molecules act as danger signals to the immune system.
Okay, like a warning.
Yeah, like an alarm. It alutes the immune system to the threat and activates various immune cells, dendritic cells, T cells, and then those cells can travel throughout the body, targeting any remaining cancer cells.
Even ones that haven't been irradiated.
Exactly. So it's like those dying cancer cells are sounding the alarm, calling in the immune system for backup.
That's amazing.
Yeah.
And is there evidence this is actually happening?
There is, in both humans and dogs.
Okay, so radiation can prime the immune system to attack cancer cells. Are there any specific examples of how this is being studied in dogs?
Oh, absolutely. One area of research is looking at the effects of radiation on the tumor microenvironment.
The tumor microenvironment. What was that again?
Think of it as the ecosystem that surrounds the tumor. It includes all the cells and molecules present in that area. Immune cells, blood vessels, all kinds of signaling molecules.
Okay, so the tumor's neighborhood.
Exactly. And it turns out that the tumor microenvironment, it can have a huge impact on how well cancer therapies work.
So how does radiation fit into that?
Well, radiation can actually alter the tumor microenvironment in a couple of ways. It can damage those blood vessels, make it harder for the tumor to receive the nutrients and oxygen it needs to grow, and it can also increase the production of certain molecules that attract immune cells to the tumor site.
So like cutting off the tumor supply lines and then sending out invitations to the immune army.
Perfect analogy. And there's growing evidence that these changes to the tumor microenvironment, they can actually make immunotherapy even more effective.
Makes sense. So the radiation sets the stage.
Exactly.
And then the immunotherapy comes in and finishes the job.
That's the idea. And there have been some really interesting studies in dogs showing this. One looked at the effects of radiation on the immune response in dogs with soft tissue sarcomas.
Okay, and what did they find?
They found that radiation increased the expression of certain immune-stimulating molecules in the tumor microenvironment, made those tumors more vulnerable to attack by the immune system.
Okay, so they're making the tumors more visible.
Exactly. And that can really change the game when it comes to immunotherapy.
So fascinating. So we've talked about immunogenic cell death, changes to the tumor microenvironment. What other ways are researchers exploring to harness this power of radiation in immunotherapy?
One area that's generating a lot of buzz is spatially fractionated radiation therapy.
Spatially fractionated. Didn't we talk about that earlier in the context of those lattice therapies?
You have a great memory. We did touch on it, but it's such an important area and things are moving so fast that it deserves a deeper dive.
Okay, I'm ready. Remind me what spatially fractionated radiation therapy is all about.
It's about delivering radiation in a more precise and targeted manner, not just blasting the entire tumor with a uniform dose.
So more like a scalpel than a sledgehammer.
Perfect analogy. And there are different ways to achieve this. One way is called GRID therapy.
GRID therapy.
It's where the radiation is delivered in a grid-like pattern
Okay.
with higher doses to specific points within the tumor. And you try to spare the surrounding healthy tissue.
So kind of like a checkerboard.
Yeah, you could think of it that way. And this approach, it's very good at killing those cancer cells while minimizing damage to healthy tissue.
Makes sense. But how does this tie into immunotherapy?
Well, there's evidence that spatially fractionated radiation therapy, it can actually enhance the immune response to the tumors.
Really? So being more precise with the radiation actually makes immunotherapy more effective?
That's what the research is showing, and there have been some very promising studies in dogs supporting this idea.
Tell me about them.
One study looked at the effects of GRID therapy in dogs with these large soft tissue sarcomas, and they found that it was very well tolerated. And it led to a decrease in certain inflammatory molecules, ones that can suppress the immune response.
So by reducing those inflammatory molecules, it's essentially making the tumor environment more immune-friendly.
Exactly. And it suggests that GRI therapy, it not only kills cancer cells directly,
But it also makes it easier for the immune system to attack the tumor.
Precisely.
Okay, so JRID therapy is showing some promise. What about other types of spatially fractionated radiation therapy?
Well, there's also lattice radiation therapy.
Lattice. How is that different?
It uses even more precise targeting than GRID therapy.
Oh, wow. And how does lattice radiation therapy impact the immune response?
Well, exciting preclinical data shows that lattice radiation therapy can upregulate certain immune-stimulating pathways in the tumor.
So it's basically sending a wake-up call to the immune system.
Exactly. And this increased immune activation can lead to a more effective attack on the tumor.
So it's not just about killing the cancer cells directly. It's also about making sure the immune system mops up any stragglers.
That's the key. And spatially fractionated radiation therapy seems to be very good at both.
Amazing. So we talked about how radiation can trigger immunogenic cell death, alter the tumor microenvironment, and how this spatially fractionated radiation therapy can really enhance the immune response. Are they also looking at combining radiation with other types of immunotherapy?
Absolutely. That's actually one of the hottest areas of research right now. There's a growing understanding that radiation and immunotherapy, they work best when used together.
Oh, that makes sense. Like they enhance each other's effect.
Exactly. They work synergistically, more than the sum of their parts. And there have been some really promising studies in dogs demonstrating this synergy.
Oh, like what?
Well, one study combined radiation with OX40 agonists and TLR agonists in dogs with a variety of different cancers. Remember those?
Yes. OX40 agonists activate T cells, and TLR agonists activate toll-like receptors, which are part of the innate immune system.
That's right. And they found that this triple combination, radiation plus those two immune stimulants, led to a really significant depletion of regulatory T cells within the tumors.
Regulatory T cells. Aren't those the ones that actually suppress the immune response?
They are. They're like the peacekeepers of the immune system. But in the context of cancer, you actually want the immune system to be as aggressive as possible.
So getting rid of them is a good thing.
It can be, yes.
So this triple combination therapy basically removed the brakes and unleashed the full power of the immune system.
That's the idea, and it's a very promising approach, and it's being explored further in clinical trials.
Any other cool combos being tested?
Oh, yeah. There's also been some really encouraging research combining radiation with IL-2 fusion cytokines, those genetically engineered proteins that stimulate the immune system.
And how's that working?
Well, a study using this approach in dogs with metastatic osteosarcoma and melanoma found that it was safe and feasible. And they actually saw some signs of efficacy.
Wow, that's awesome. It really seems like the future of cancer treatment is going to involve a lot of these combination therapies, you know?
Absolutely. And research in dogs, it's helping us figure out the best way to combine these therapies to get the best results.
It is so amazing to think that all of this research is happening in dogs and directly informing the development of these new cancer treatments for humans. It's mind-blowing.
It truly is remarkable, and it shows the power of comparative oncology.
Comparative oncology, what's that?
It's the study of cancer across different species. So by studying cancer in dogs, we're not only helping them, but also gaining invaluable insights that could lead to breakthroughs for humans as well.
Okay, so we've covered a lot of ground on radiation therapy. What other breakthroughs are happening in the world of canine cancer immunotherapy?
One area that I find particularly exciting is the development of new tools and techniques for immunomonitoring.
Right, we touched on that earlier, how important it is to track the immune system's response to therapy. But I'm eager to learn more about the specifics.
Imagine being able to peer inside a tumor and see exactly what kinds of immune cells are there and how active they are.
Wow, that would be incredible. Is that even possible?
It is becoming possible, thanks to some amazing technological advances.
Okay, like what?
One of the most powerful tools is flow cytometry. It lets you identify and quantify the different types of cells in a sample, including immune cells.
Oh, cool. So you can actually see what's happening inside.
Exactly. You can see which types of immune cells are present, how many there are, how active they are.
That's incredibly valuable. So how are they using flow cytometry for canine cancer immunotherapy?
Well, for one thing, it's helping researchers understand how different immunotherapies are actually affecting the immune system. They can track changes in the number and types of immune cells in, say, the blood or tumor tissue of a dog who's undergoing immunotherapy.
So they can basically see how the immune system is responding to the treatment.
Exactly. And that information can then be used to optimize those treatment strategies, to get better outcomes.
That makes sense. So it's like a feedback loop.
It is, and it can also help identify potential biomarkers, those measurable indicators that can predict which dogs are most likely to respond to immunotherapy.
Oh, okay. So, like, if you see this, it's a good sign.
Exactly. And they're finding that certain immune cell populations are associated with better responses to immunotherapy.
So if a dog has a lot of these specific cells, it's a good indicator they'll benefit from treatment.
That's the idea. This kind of information helps personalize those treatment strategies and improve the chances of success.
Wow. So flow cytometry is really important for this research. Are there any other technologies they're using for immunomonitoring in dogs?
Absolutely. Another powerful tool is RNA sequencing.
RNA sequencing. Didn't we talk about that earlier?
You did. It's a versatile tool.
Okay, so remind me again how it works and how it's being used in the context of immunotherapy.
So remember DNA, the genetic blueprint?
Right.
Well, it's RNA that actually carries out those instructions. RNA sequencing lets you analyze those RNA molecules in a sample, and it tells you which genes are being expressed.
Oh, right. So like which genes are turned on.
Exactly. And for immunotherapy, RNA sequencing is really useful for studying those gene expression patterns of immune cells. So it's telling you which genes are being activated in immune cells as they respond to immunotherapy.
Wow, that's incredible. You're able to see how the code is changing.
That's a great way to put it. And they're already finding some interesting stuff in dogs.
Oh, yeah. Like what?
Well, one study compared the gene expression patterns of T cells from dogs and humans after they stimulated them with these immune activating molecules.
What did they find?
They found both similarities and differences in how the T cells from the two species responded.
So we're similar, but different.
Exactly. And knowing those differences, it's really important for helping researchers interpret the results of canine immunotherapy trials and translate those findings to human patients.
That makes sense. Okay, so RNA sequencing is giving some really valuable insights into the canine immune system. What other technologies are they using for immunomonitoring?
Another one is cytokine measurements.
Cytokines. Those are those signaling molecules that immune cells use to talk to each other, right?
You got it. They're basically the language of the immune system.
So how are cytokine measurements used in canine cancer immunotherapy?
Well, by measuring the levels of different cytokines in the blood or tumor tissue.
Of dogs undergoing immunotherapy?
Exactly. You can actually get a sense of how active that immune system is.
So you're eavesdropping on their conversations.
You could say that. And this type of analysis, it can be really useful for getting information about how well the therapy is working and whether the immune system is actually mounting a strong attack against the tumor.
Okay, so we've got flow cytometry, RNA sequencing, and cytokine measurements.
Yeah.
Anything else in the immunomonitoring toolbox?
Oh, yeah. Another really useful technique is nanostring analysis.
Nanostring analysis.
It allows you to measure the levels of very specific RNA molecules very precisely.
So it's kind of like a more targeted version of RNA sequencing.
Exactly. And it's really helpful for doing something called immune profiling of tumors. Well, you're analyzing the types and numbers of immune cells in the tumor microenvironment.
Oh, wow. So you can get a really detailed snapshot of what's happening inside the tumor.
Exactly. And then you can use that information to adjust those treatment strategies and improve outcomes.
Amazing.
What else?
Well, one more area that's really exciting is the development of techniques to assess TCR clonality.
TCR clonality. Now, that's a new one. What's that all about?
Okay, so remember T cell receptors, or TCRs?
Yes. They're the proteins that allow T cells to recognize specific antigens, kind of like their weapons.
Exactly. Well, TCR clonality refers to the diversity of TCRs in a sample. Basically, the more diverse the TCR repertoire,
The more weapons they have.
exactly, the better equipped that immune system is to recognize and attack a wider range of invaders.
That makes sense. So how does that relate to immunotherapy?
Well, by analyzing TCR clonality, you can get a sense of how well the immune system is responding to immunotherapy.
So if you see the TCR repertoire getting more diverse, that's a good sign. It can be. It suggests that the immune system is getting more activated and becoming more capable of recognizing and attacking those cancer cells.
Okay, I get it. So what kind of techniques are used to assess TCR clonality?
There are a couple approaches. One is bulk RNA sequencing.
Which we talked about earlier.
Yes, it can give you a good overview, but the most cutting-edge approach is single-cell TCR sequencing.
Single cell. So you can analyze each individual T cell.
Exactly. And it gives you an incredibly detailed view of the immune response. You can see how those individual T cells are responding to therapy.
That's remarkable. It sounds incredibly powerful. Are they using single cell TCR sequencing in dogs?
They are. One study used it to create this incredibly detailed atlas of the immune cells that are present in the blood of dogs with osteosarcoma.
An atlas. That sounds really comprehensive.
It is. It's like having this roadmap of the immune system in dogs with that specific type of cancer.
So what did they find?
Well, one of the really interesting things they found was that certain types of myeloid-derived suppressor cells.
Myeloid-derived suppressor cells.
Mm-hmm.
Those are the immune system traders, right?
Got it. Well, those cells were much more abundant in the tumor-bearing dogs compared to healthy dogs.
So their presence makes it harder for the immune system to fight the cancer.
It does. And a lot of research now is actually focused on targeting these suppressor cells, either eliminating them or blocking their suppressive activity to kind of unlock the full potential of the immune system.
Wow, that's really cool. So they're finding new targets for treatment.
Exactly.
So single-cell sequencing is revolutionizing our understanding of the canine immune system. What else can it do?
Well, one of the most exciting applications is in single-cell TCR sequencing, which we talked about earlier, right?
Yes. Analyzing those T-cell receptors at the single-cell level, it's pretty mind-blowing.
It is. It gives us this incredibly fine-grained view of the immune response. You can actually see how individual T-cells are responding to therapy.
Okay, but why is that so important? What can we learn from that?
Well, for one thing, it can help us understand how different T-cell subsets are responding to immunotherapy.
Oh, right. They're all different kinds of T-cells, right? Helper T-cells, cytotoxic T-cells.
Regulatory T-cells, all sorts. And each one plays a unique role. And by analyzing those TCRs of those different subsets, you get this much more complete picture of how the immune system is responding.
It's like seeing the individual players on the team and understanding how they're each performing.
Exactly. And this is crucial for developing more effective immunotherapies.
So is single-cell TCR sequencing being used in dogs with cancer?
It is. One study used it to analyze the T-cell response in dogs with melanoma, and these dogs are undergoing immunotherapy.
Okay, what did they find?
They found that the dogs that responded well to the treatment,
They had
a more diverse TCR repertoire than those that didn't respond as well.
So diversity is key. The more diverse the receptors, the better the immune system can fight the cancer.
Exactly. And this finding suggests that TCR clonality could actually be a valuable biomarker for predicting how well a dog will respond to treatment.
So they could use this technology to identify the dogs who are most likely to benefit.
That's the idea. And it could really help to personalize those treatment strategies and improve outcomes.
This is incredible. We can analyze the immune response at such a granular level. It's just remarkable.
It is, all thanks to these huge advances in technology in recent years.
It really makes you wonder what other secrets are hidden within the immune system, just waiting to be discovered.
That is the million-dollar question. And I think that with all this research and innovation, we're going to be unlocking those secrets and developing even more effective immunotherapies for dogs and humans.
This has been an incredible journey. I can't believe how much we've learned about canine cancer immunotherapy.
Me too. And it's a field that's moving so fast. It's really exciting to see.
I know. Okay, so we've talked about the basics of the immune system, all these cutting-edge technologies. I have a feeling we're still just scratching the surface.
Oh, you're absolutely right. There are still many unanswered questions. But all of this research in dogs, it's paving the way for some incredible discoveries.
Well, I'm ready to dive even deeper. What other fascinating things can you tell us about canine cancer immunotherapy?
Well, one area that is particularly exciting is the development of new strategies to enhance immunotherapy's effectiveness.
Enhance the effectiveness. So we're talking about making these already powerful therapies even better.
Exactly. Researchers are always trying to improve these therapies, fine-tune them, make them even more potent.
That makes sense. If we can make the immune system fight cancer even more effectively, that's a huge win.
It would be, and there are a lot of different approaches being explored.
Like what?
Well, one approach is combining different types of immunotherapy. We talked about that earlier with radiation therapy, but it's being explored for other types of immunotherapy as well.
Okay, so instead of just using one type of immunotherapy, you might use 2 or even 3 different types together.
That's right. You're hitting it from all angles, and they're seeing some promising results with this in dogs already.
Really?
There was one study where they combined a cancer vaccine with an immune checkpoint inhibitor
In dogs with?
melanoma. And they found that this combo was much more effective at shrinking tumors and extending survival times than either treatment by itself.
Wow, that's incredible. So 2 is better than 1.
In some cases, yes.
Okay, so that's one approach. What else?
Well, another approach is personalizing immunotherapy based on the individual patient's tumor and immune profile.
So tailoring the treatment.
Exactly. And that's where all those immunomonitoring tools we talked about come in.
Right. All that information they gather can help them choose the right treatment.
It's all about understanding those details so that you can select the immunotherapies that are most likely to be effective for that particular dog.
That's incredible. It's like custom design treatment.
That's the goal. And it's an exciting area of research, one that has a lot of potential.
Okay, so we've got combining different immunotherapies and personalizing immunotherapy. What else is on the horizon?
Well, researchers are also exploring new ways to deliver immunotherapy.
What do you mean new ways to deliver? Don't they just give an injection?
Well, traditionally, most immunotherapies are delivered systemically, either intravenously or subcutaneously.
Right, so that means the treatment is injected into the bloodstream or under the skin, and then it circulates throughout the body.
Exactly. But researchers are now exploring ways to deliver that immunotherapy directly to the tumor site.
Directly to the tumor. Why would that be better? Wouldn't that make it harder to reach cancer cells that have spread to other parts of the body?
That's a great question, and it's definitely something researchers are looking into. But the thinking is that delivering the immunotherapy right to the tumor could make the treatment more effective.
How so?
Well, by increasing the concentration of the treatment right where it's needed most.
So instead of having it travel through the whole body, you just concentrate all of its power right at the source. Makes sense. But how do you actually do that? How do you get the treatment to the tumor?
Well, there are a few different approaches. One is to just inject the immunotherapy directly into the tumor.
Okay, so just like a shot right into the tumor.
Pretty much. Are there any other options?
Well, that seems pretty straightforward. Are there any other options?
Another approach is using nanoparticles to deliver that immunotherapy.
Nanoparticles, those are those tiny microscopic particles, right?
That's right. And these nanoparticles can be engineered to specifically target those tumor cells.
Oh, wow. So they're like microscopic delivery trucks.
Exactly.
Carrying the immunotherapy right to the tumor. That's incredible.
And because nanoparticles are so small, they can penetrate really deep into that tumor tissue, delivering that treatment exactly where it needs to go.
Wow, that's amazing. Are there any other ways to deliver immunotherapy directly to those tumors?
There is some really cutting-edge research using oncolytic viruses.
Oncolytic viruses. Did we talk about those earlier?
We did. Those are the viruses that are engineered to infect and kill cancer cells.
Oh, right. And they can trigger immunogenic cell death. So you're basically using these viruses as a double whammy.
Right.
Killing cancer cells directly and then calling in the immune system for backup.
Exactly. And they can also be used to deliver other types of immunotherapy.
Oh, really? Like what?
Like cytokines or checkpoint inhibitors.
So they're kind of like Trojan horses.
Yes.
Sneaking into the tumor and then releasing all these immune-boosting agents.
Precisely. And this approach is showing a lot of promise in those preclinical and clinical trials.
This is also fascinating. So we've talked about combining those different immunotherapies, personalizing immunotherapy, and then exploring all these new delivery methods. What other strategies are they using to try to make immunotherapy even more effective?
Well, there's a growing interest in understanding the role of the microbiome in cancer immunotherapy.
The microbiome, that's all the bacteria and microorganisms that live in our gut, right?
That's right. And we're learning more and more that it can really impact our immune system.
Okay, but how does that relate to cancer and immunotherapy?
Well, there's evidence that the composition of our microbiome can actually influence how well a patient responds to immunotherapy.
Wait, are you serious? So the bacteria in our gut can affect how well our immune system fights cancer?
That's what the research is showing, and it's opening up this whole new avenue for improving cancer treatment.
Wow, that is incredible. What's the connection there between the microbiome and the immune system?
Well, the bacteria in our gut, they're in constant communication with our immune system, helping to shape it and train it.
So the bacteria are like teachers for our immune system.
You could say that, and a healthy, diverse microbiome can help to create a really strong and balanced immune system.
One that's better at fighting off infections and diseases.
Including cancer.
So if you have like an unhealthy microbiome, could that weaken your immune system and make it harder to fight off cancer?
That's what the research is suggesting.
Wow, that's incredible. Okay, so how does this all relate to canine cancer immunotherapy?
Well, just like humans, dogs have a microbiome, and it's really important for their immune health. And researchers are now looking at ways to manipulate that microbiome to make cancer immunotherapy even more effective in dogs.
Manipulate the microbiome. How do you even do that?
Well, one approach is using probiotics.
Probiotics, those are those live bacteria that are good for your gut, right?
You got it, the good bacteria you find in yogurt and things like that.
And those can boost the immune response to cancer.
There's growing evidence that certain probiotics can, and they're actually seeing some really promising results in dogs using probiotics to enhance the effectiveness of cancer vaccines.
Wow, that is amazing. So you're basically giving the immune system a little extra help
Exactly.
in the form of helpful bacteria. That's incredible. Okay, so probiotics are one way to manipulate the microbiome. What are some other methods?
Another approach is fecal microbiota transplantation, or FMT.
FMT. That sounds a little intense.
It might sound a bit unconventional, but it can be a very effective way to change the composition of the microbiome.
So how does it work?
Well, it involves transferring fecal matter from a healthy donor into the recipient's gut.
Oh, wow. So they're basically getting a whole new set of gut bacteria.
Yes, and it can have a big impact on the recipient's immune system.
Okay, and how is FMT being used in dogs with cancer?
Well, there have been some small studies suggesting that FMT can actually enhance the response to those checkpoint inhibitors.
Oh, wow. So they're seeing some positive results.
They are, and it's an area with a lot of potential for the future.
This has been an incredible deep dive. I can't believe how much is happening in this field. It's absolutely amazing.
It is a really exciting time to be following this research, and the progress that's being made is benefiting both dogs and humans.
It really is one health in action. It's amazing to think that our dogs are playing such an important role in the fight against cancer.
I completely agree, and I think as we continue to unravel the mysteries of the canine immune system, we're going to see even more breakthroughs in the years to come.
It makes you wonder what other amazing discoveries are just around the corner.
That is the million-dollar question, and I have no doubt that with all the amazing research and collaboration going on, we're going to see even more progress in the fight against cancer for both dogs and humans.
This has been an amazing journey. Thank you so much for taking us along with you.
It's been my pleasure, and as always, from Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.